Good morning all of you. Today we are going to discuss the biochemistry portion of the Grand Test 179. Now let's start with the discussion. Let's take question number 1. Identify the given test for proteins. Now whenever this type of question comes in the exam, first of all you have to recall all the tests which are given by the proteins. Now proteins, as all of you know, proteins are made of they are basically the polymers of amino acids. Amino acids which have a Zwitter ion structure. Now what do you mean by this Zwitter ion structure? They are amphoteric in nature. Means if on one hand they can behave as an acid, on the other hand they can behave as the base. Now whenever we have to detect the presence of proteins, practically there are two types of tests. One is qualitative. Qualitative means by seeing the quality of the color, we can say this protein consists of this amino acid. Second is quantitative. Quantitative means we have, and it is clear from the word quantity means we are checking how much amount of protein is present in that. Now qualitative mein there are two confirmatory tests which are, one is biuret, which is a confirmatory test for every protein. Second is ninhydrin test. Ninhydrin test is again a confirmatory test for every protein. Then there are certain other tests which basically give color on the presence of certain amino acids in it. Just for example, xanthoproteic test is there, which is again a very important test. Sakaguchi test is there. Hopkins-Colles test is there. Now let's take over the description of these tests. Biuret, as it is clear from the word itself, biurea means whenever two molecules of urea are heated in the presence of copper sulfate, they are going to give a coordination compound which is mainly blue in color. Since you know that every protein is formed of a peptide bond, the main requisite for a protein to give a positive biuret test is it should have more than 2 peptide bonds. This is very, very important. If a protein has more than 2 peptide bonds, automatically it is going to give a positive biuret test. And if a protein has less than 2 peptide bonds, it is not going to give a positive biuret test, and that will be known as a biuret product. Ninhydrin it is going to give purple color, and the principle behind this ninhydrin is it is simply decarboxylation of the amino acid, and isme we have to boil the solution. So biuret and ninhydrin are considered to be the two confirmatory tests. Xanthoproteic test is for the aromatic amino acids. Like phenylalanine is there, tyrosine is there, tryptophan is there. Sakaguchi test is basically for the guanido group. Guanido group, which is a characteristic feature of arginine. So arginine gives a positive Sakaguchi test. Hopkins-Colles test is for tryptophan, as tryptophan has a indole ring in it. Similarly, there are other tests also, like lead acetate test. This lead acetate test is for sulfur containing amino acids, which is cysteine and methionine. Now here one thing I'll point out, cysteine will give a positive lead acetate test, methionine cannot give a positive lead acetate test. The reason behind this is, in this methionine, it has one methyl group. So this lead, the alkali cannot break this linkage. So lead acetate is given by cysteine, which is again a sulfur containing amino acid. Then Millons test. Millons test is a test, specific test for tyrosine. So these are the different type of qualitative tests. Then another segment was the quantitative one. Quantitative means we are predicting the amount of protein present in it. Quantitative test is mainly the biuret test, or you can say biuret test is the only test which is qualitative as well as quantitative. For example, if we have to assess the amount of protein in a vaccine, we carry out this biuret test. There are other tests also, like Lowry's test is there, Bradford test is there. These are what? These are the quantitative tests which are basically based on Lambert-Beer's law. This is again a very, very important question which is mainly asked in the exam. These proteins or the amino acids absorb at a particular wavelength. They give a absorption spectra, and by that we calculate the amount of amino acid or a protein present in the sample. Now here if you look at the question, you can see the other different tests are Barfoit's test, Paton-Koffer test, biuret test, Moll's test. सबसे पहले the question arises, if we are asking the given test for proteins, all other tests are not for proteins. The option is automatically it is biuret. But you should know the other tests also. Now let's take up Barfoit's test first of all. Now see, Barfoit's test and Barfoit's and Mollish, these are the tests for carbohydrates. Mollish test is considered to be the confirmatory test for every carbohydrate. Every carbohydrate will give a positive Mollish test. Barfoit's test distinguishes between mono and disaccharides. Simply inodiol formation is there if precipitation appears in a second. If precipitates appear in a second, it is a monosaccharide. Delayed precipitation predicts it is a disaccharides. So Barfoit's and Mollish are for carbohydrates. Paton-Koffer test is for bile acids. There is another test, Hayes test, for bile salts. So here the option is biuret test, which is a confirmatory test for proteins, one thing, and second is it is a qualitative as well as a quantitative test. This is very, very important. Then let's switch over to next question. The given feature is of which storage disorder. Now here you can see very well from the picture only, there is calcification of the adrenal glands. And whenever we see this type of picture, calcination of adrenal glands is wholly solely a picture of Waldmann's disease. This Waldmann's disease is due to the absence of Waldmann's disease, cholesterol ester hydrolase. Cholesterol moves in free form also. Cholesterol moves in esterified form also. Here I would like to bring one more thing into picture. Esterification of cholesterol is carried out by a very, very important enzyme which is LCAT, lecithin cholesterol acyl transferase, and this acetylation takes place at second position, one thing. Deficiency of this enzyme leads to Norham's disease. Now here the question is, there is simple calcification of the adrenal glands. There is accumulation of fatty acid crystals around the adrenal gland. This is mainly due to the absence of this cholesterol ester hydrolase. Other options are sphingolipids. Sphingolipids are nothing but the type of phospholipids which have a unsaturated carbon atom, and the alcohol here is sphignol. Sphingolipids is a characteristic disorder which is mainly due to the deficiency of, there are so many disorders associated to it, like Tay-Sachs disease is there, then Gaucher's disease is there, which is due to the deficiency of hexosaminidase A and B. So sphingolipidosis is mainly due to this, and another option is Niemann-Pick disease. This is again very important. This Niemann-Pick disease is due to the deficiency of the enzyme sphingomyelinase. There is accumulation or there is enlargement of the fatty layer. So you can say here, simple, by just looking at the picture, we can say the answer is Wallmann's disease, which is due to cholesterol ester hydrolase. Then switch over to next question. A patient has total cholesterol 300. What we have to find out LDL. Simple. The formula for calculating LDL is total cholesterol minus HDL cholesterol minus triglycerides over 5. Now here the concentration of total cholesterol is 300, 150 and 25. 300 150 150 is TG and HDL is 25. HDL is 25. Simple, we calculated 30, 300, 30, 25. The answer would be 245. This is how we calculate the LDL. Similarly, we can calculate VLDL also. VLDL, very low density lipoproteins, are simply calculated by TGs divided by 5. The amount of total triglycerides divided by 5 will give the concentration of the VLDL. This is how we calculate it. Is it clear? Now, next question. Which among the following amino acids absorb UV light? Now see, there are so many questions which have been asked mainly on the absorption pattern of the metabolites. Now here what we are going to, let me see, absorbance depends upon Lambert-Beer's law. They were basically the two scientists who gave the concept of principle of calorimeter or analyzer, we can say. Now what happens here is, it is directly proportional. More the absorbance is directly proportional to the amount of the metabolite in that unknown substance. More the absorbance, more will be the concentration of that unknown substance. Whether we have to estimate glucose, whether we have to estimate urea, whether we have to estimate uric acid, proteins, whatever we have to estimate, they are going to absorb at a particular wavelength. If the concentration of these metabolites would be more, automatically there will be more absorbance and more optical density. Now, there are certain metabolites which absorb at UVA region. There are certain metabolites which absorb in visible region. So, proteins में the aromatic amino acids basically, like aromatic amino acids are those which have a benzene ring in them. For example, phenylalanine, tyrosine and tryptophan. These three are the three aromatic amino acids which always absorb at UV wavelength. Similarly, nucleotides like DNA and RNA, they also absorb at UV light. They absorb at roughly 280 nanometers. 260 to 280 nanometers we can say, and this absorption is mainly due to the presence of the bases in them. Because if we see the structure of DNA and RNA, they have hydrogen bond. If we talk about DNA, it has hydrogen bond between the bases, phosphodiester bond between sugar and phosphate, and glycosidic bond between the sugar and the bases. The main difference is only in the composition of the bases, rest is same. So bases it absorbs at 260 to 280 nanometers. Similarly, you can say others. If we say porphyrins absorb roughly at 440 nanometers, 440 or 400 nanometers, and they give a solid band kind of appearance. So whenever we talk about UV range in case of amino acids, it's the aromatic amino acids which are absorbing at the UV range. Is it clear? Next. Next question. Now see, here if we see which of the following describes the function of the enzymes or cofactors, whenever we talk about this, automatically the answer is vitamin K, which is also known as anti-hemorrhagic factor. Vitamin K, the major role of this vitamin K is, the active form of vitamin K is phylloquinone, and it basically helps in the major function of this vitamin K is carboxylation of the clotting factors. This is the major function of vitamin K. Carboxylation means it helps in attachment of carbon dioxide to, basically what happens is this prothrombin is converted into thrombin, fibrinogen into fibrin, and finally a clot is produced. This prothrombin has glutamic acid at the side chain, and this glutamic acid is basically carboxylated in the presence of this vitamin K. This is basically one cycle. This vitamin K, actually what happens is, it is reduced. When it is converted to its oxidized form, there is gamma carboxylation of prothrombin residue, and this oxidized is converted into reduced form after that, so that it remains the same, and that is what is the idea of this functioning of vitamin K. This gamma carboxylation is basically the post-translational modification. Post-translational modification. This is a very, very important question. So the clotting factors, basically, to be more precise, this prothrombin, which has glutamic acid as one of the side chains, is carboxylated at the gamma position with the help of this vitamin K. So the answer to this question is carboxylation of glutamic acid residues. Now next question. HMG-CoA is formed during the metabolism of all except. Now see, HMG-CoA. What is this HMG-CoA? This HMG-CoA is beta hydroxy, beta methyl gluteryl CoA. This is the very important intermediate which is produced during cholesterol biosynthesis, one thing, and in ketogenesis or ketone bodies synthesis. This is very, very important, and there are so many questions which have been asked on this intermediate HMG-CoA. Now see, let's discuss this. Actually what happens is, if we take the synthesis of cholesterol as well as ketone bodies till the formation of HMG-CoA, it is the same. Just for example, what happens is, two molecules of acetyl coenzyme A acetyl-CoA, they combine to form acetoacetyl-CoA. This acetoacetyl-CoA then finally combines with one more molecule of acetyl-CoA, forming this HMG-CoA. Now, if this HMG-CoA undergoes reduction, it forms mevalonate, or it leads to the formation of cholesterol, and if it undergoes splitting in the presence of lyase enzyme, it forms the first ketone body, which is acetoacetate. Then automatically the rest of the two ketone bodies are produced. So we can say this HMG-CoA is the most important intermediate which is formed by cholesterol, by ketone bodies. The other two options are leucine. Leucine is what? Leucine is a potent ketogenic amino acid. This is very, very important. Ketogenic amino acid means ketogenic. Genesis is formation of a keto group. If a particular amino acid is generating that, it is a ketogenic one. There are glucogenic amino acids also. Glucogenic are what? Which are related to the formation of this glucose. So here leucine can also be it is a ketogenic amino acid, so it will be finally degraded to form this HMG-CoA. So the option left is bile acids. So the answer to this question would be HMG-CoA is formed during metabolism of all except bile acids. Is it clear? Then next, xanthuranic acid in the urine is suggestive of what? Now see, xanthuranic acid is basically excreted, or you can say this xanthuranic acid is xanthuranic acid. It is related with the metabolism of tryptophan. Tryptophan, which has a indole ring attached to it. Actually, tryptophan is classified under aromatic amino acid as well as heterocyclic amino acids. Why aromatic? Because it has a benzene ring in it. Due to the presence of this indole ring, it is also known as a heterocyclic amino acid. Now, all of you know that amino acid has the metabolism based on nitrogen skeleton as well as the carbon skeleton. Nitrogen skeleton means they will be finally degraded to form urea in the end. And carbon skeleton mein they can be glucogenic and they can be ketogenic. Now see, tryptophan, once its function is over, because tryptophan is again a very important, it is an essential amino acid, because it is required for the synthesis of one thing, niacin, and it is required for the synthesis of melatonin. So these are the two very important features, products of tryptophan. Now once this tryptophan is degraded to form these, obviously metabolism is what? It has to be degraded to form these important compounds. This tryptophan is degraded via two pathways. One is this kynin-urine pathway and other pathway is the serotonin pathway. Serotonin, which is a potent vasoconstrictor, it is synthesized from tryptophan. This is again a very important question. Serotonin is the precursor of serotonin, we can say, is tryptophan. Now kynin-urine pathway plays a very important role. This tryptophan will be degraded to form N-formyl kynin-urine. This N-formyl kynin-urine will then be degraded to form kynin-urine. This kynin-urine will then be converted into 3-hydroxyl kynin-urine. This 3-hydroxyl kynin-urine is converted into 3-hydroxyl anthranilate or antranilic acid. In the presence of enzyme kyninurinase, and this 3-hydroxyl anthranilate by two-step reaction finally forms nicotine, or you can say NAD, or you can say niacin. Now what happens is, this kyninurinase requires vitamin B6 for this reaction. Deficiency of vitamin B6 leads to increased concentration of kyninurine, and this is not then in the deficiency of vitamin B6, it will be converted into xanthurenic acid. So you can say in vitamin B6 deficient subjects, there is excretion of xanthurenic acid in urine. And that is what the question says. Xanthurenic acid is suggested of deficiency of which vitamin? So it is vitamin B6. Now vitamin B6, it is pyridoxine. Active form of pyridoxine is PLP. PLP is a very important coenzyme of transamination also. This is again a very important question. Transamination cannot occur without pyridoxal phosphate. This has again been asked in the exam so many times. So answer here is, due to the deficiency of B6, instead of the formation of 3-hydroxyl anthranilate, this 3-hydroxyl kyninurine will finally be converted into xanthurenic acid, and there is excretion of this in urine. Now next question. Succinate dehydrogenase is inhibited by. Now see, succinate dehydrogenase, or commonly it is known as SDH. It is an enzyme of TCA cycle. Dehydrogenation, removal of hydrogen is there. So it converts succinic acid to fumaric acid. Since it is linked with dehydrogenation, automatically there is dehydrogenation. This enzyme is very, very important. There are important points about this enzyme. First important point about this enzyme is this is the only enzyme of TCA which is present in the inner mitochondrial membrane. Because all of you know that TCA cycle takes place in the matrix of mitochondria. This SDH is the only enzyme which is present on the inner mitochondrial membrane. One thing. Second is it has a very low redox potential. Now since if it has a very low redox potential, it means that it always generates, you can say, 1 ATP lesser than what NAD gives. That is why we say one molecule of FADH2 always gives 1.5 ATP molecules. In contrast to what NAD gives. NAD gives 2.5 ATP molecules. So it has one molecule of ATP lesser. Why? Because of low redox potential, and it cannot contribute to PMF, or you can say proton motive force. This is again a very, very important question. An inhibitor of this is malonate. Malonate is a competitive inhibitor of succinic acid or SDH. Competitive inhibitor means there is a competition. Malonate resembles succinic acid in structure. So what happens is, if malonate binds, automatically there won't be dehydrogenation, or fumaric acid won't be produced, or the TCA cycle will suffer. Another very important feature which I would like to bring in competitive inhibition is, in this competitive inhibition always there is change in the Km. Km decreases because the affinity, Km increases because the affinity of the enzyme with the substrate decreases. Because there is a competition between the inhibitor and the substrate. It's like this. The inhibitor binds with the active site of the enzyme. So Km increases, but Vmax is unchanged. So this malonic acid or malonate is a competitive inhibitor of succinate dehydrogenase, which is the only enzyme of TCA which is present in the inner mitochondrial membrane. Now next, a 2-year-old intellectually disabled child is having blue eyes, blonde hair and fair skin. He also has a peculiar body odor. What is the diagnosis? Now whenever this, again when this type of question comes, see blue eyes, first of all let's look at the option MSUD, maple syrup urine disease, isovaleric aciduria, phenylketonuria and Canavan's disease. Whenever we talk about blue eyes or fair skin, now see, it is a characteristic feature of phenylketonuria. Phenylketonuria, which is due to the deficiency of enzyme phenylalanine hydroxylase. This enzyme is responsible for the conversion of phenylalanine to tyrosine. Phenylalanine is considered to be an essential amino acid. Why it is essential? Because it is required for the synthesis of tyrosine. This tyrosine is a precursor of so many important compounds, whether it is triiodothyronine, whether it is tetraiodothyronine, whether it is melanin, whether it is epinephrine and norepinephrine, or you can say the catecholamines. The precursor of catecholamines, thyroid hormones, melanin, is tyrosine, which is generated from phenylalanine in the presence of phenylalanine hydroxylase. This reaction requires molecular oxygen, NADPH. These are the important components of this. Without this, the reaction cannot proceed. Now what happens is, if this enzyme is deficient, automatically there won't be production of tyrosine, accumulation of phenylalanine will be there in the blood, and there will be excretion of phenylalanine in urine, leading to phenylketonuria. And this phenylalanine can be converted into phenylacetic acid, or it can be converted into phenylpyruvic acid. Now see, the child is complaining of what? Blue eyes, fair skin. That means there is no synthesis of melanin due to the deficiency of this enzyme. It has to be phenylketonuria. Now, let's take the other options also. Say MSUD. MSUD is what? Simple maple syrup urine disease. It is mainly due to the branched chain amino acids, and branched chain amino acids are valine, leucine, isoleucine, and the deficient enzyme is alpha keto acid dehydrogenase, or it is decarboxylase or dehydrogenase. Right? Here the urine starts giving the smell of burnt sugar. That is why it is known as maple syrup urine disease, due to the smell of this burnt sugar in it, and the deficient enzyme is this. There is increased concentration of the amino acids in the urine. Another is isovaleric aciduria. This isovaleric aciduria is mainly due to the deficiency of iso-valeryl coenzyme A dehydrogenase. Here basically what happens is, leucine, which is a potent ketogenic amino acid, it is degraded to form alpha keto isocaproic acid. This alpha keto isocaproic acid is converted into isobaric CoA. This isobaric CoA undergoes dehydrogenation in the presence of this enzyme, forming beta methyl crotonyl CoA. And this beta methyl crotonyl CoA by different steps will be finally converted into, it will be degraded to form carbon dioxide and water. Now deficiency of this enzyme will lead to what? Accumulation of this isobaric CoA, leading to excretion of this isovaleric acid in urine, causing isovaleric aciduria. Major symptoms of this is, the patient has sweaty feet odor, stupors, and the person can go in coma also. But again, there is no concept of blue eyes here, and this is mainly due to the deficiency of enzyme isobaric coenzyme A dehydrogenase. Phenylketonuria. Then is Canavan's disease. This Canavan's disease, Canavan's disease is due to the deficiency of enzyme Amino acylase 2. This is again basically an autosomal recessive disorder, and the symptoms are paralysis is there, blindness, or seizures may occur. ठीक है, deficient enzyme is this. Now here if we look at the question mate is shown very clearly. You can see the picture also. It has to be phenylketonuria, which is due to the deficiency of phenylalanine hydroxylase, the enzyme required to convert phenylalanine to tyrosine, which is a precursor of melanin. Deficiency leads to lack of the synthesis of melanin, leading to no change in the skin color. The next question is, the OSA zones given in the diagram are respectively. Now first of all, before attempting this question, you should know what are OSA zones. See, OSA zones they are basically the crystals of carbohydrates which are reducing in nature. This is again very important. Or you can say all the reducing sugars have a tendency to form these OSA zones. Basically what happens is, we take the reducing sugar, we add phenylhydrazine hydrochloride and sodium acetate. Keep the solution overnight and automatically there will be precipitation of the crystals. So all the reducing sugars, when they combine with phenylhydrazine, they are going to form the respective OSA zones. Jaise for example, glucose is going to form glucosa zone. Fructose is going to form fructosa zone. Maltose is going to form maltosa zone. Similarly, lactose is going to form lactosa zone. And you can say sucrose, being a non-reducing sugar, it won't form any OSA zone. This is again a question which is asked in the exam. All the reducing sugars can give OSA zone except. This is a very frequent question asked. So here if we see OSA zone mein, the structure of the crystals of glucosa zone and fructosa zone will be needle shape or bundle of hay. Maltose will have sunflower petal-like structure. Lactose will have powdery puff. So by seeing the shape of the structure of the crystals, we can predict the OSA zones present. Now let's see the question. यहाँ पे you can see this is what? This is, if we see the first picture, it is hay, bundle of hay or needle-like. It is glucose. Second is powdery puff. Powdery puff is of maltose or lactose. It is of lactose. And third is, it resembles sunflower, so it is maltose. So the answer would be glucosa zone, lactosa zone and maltosa zone. Sucrose cannot form OSA zones as it is a non-reducing sugar. Rest of the reducing sugars form that. The next question, example of mixed function oxidase. Now see, what do we mean by this mixed function oxidase? Mixed function oxidase. For understanding this concept, we should know what is this oxidase. Oxidase, again it is clear from the word itself, it can catalyze the oxidation. Oxidation, or there will be loss of the electrons. If we talk about a typical biochemical reaction, it is nothing but a redox reaction in which oxidation and reduction take place simultaneously. If a substrate is oxidized, automatically something has to be there which is ready to accept those electrons, or that will be reduced. So mixed function means it is having a mixed kind of function. This oxidases is a very wide class of enzymes which has so many subtypes. Oxygenases is there, dehydrogenases is there, which is further divided into subclasses. Just oxygenases is what? It can be monooxygenase, it can be dioxygenase. Monooxygenase, again it is, देखो, oxygenases means there is incorporation of oxygen. Incorporation of this molecular oxygen, so that the substrate is oxidized. Finally, it is going to form carbon dioxide and the water. Monooxygenase means there is incorporation of oxygen, but only one molecule of oxygen is there. Mono means one. One molecule of oxygen will be there, and automatically the substrate will be oxidized. There will be a reductant present which is finally degraded to form water soluble products which are excreted in urine. Dioxygenases means there is incorporation of two molecules of oxygen. Ultimately what is happening, whether it is monooxygenase, whether it is dioxygenase, it has to convert that substrate so that finally carbon dioxide and water will be generated. This is how it occurs. Now example of monooxygenases, if you look at the options, homogenistic acid oxidase, cytochrome P450, tryptophan pyrolysin, liver enzymes. Except cytochrome P450, all come under dioxygenases. Means there is incorporation of two molecules of oxygen whenever this homogenistic acid oxidase enzyme acts. This homogenistic acid oxidase enzyme is the enzyme of homogenistic acid oxidase enzyme. Deficiency of this enzyme leads to alkaptonuria. Basically what happens is, if this homogenistic acid oxidase is not there, there won't be oxidation of this homogenistic acid. And this homogenistic acid will be finally converted to form alkapton type of or quinone type of substances. The urine turns black on exposure to air. That is what is the characteristic feature of this. So there is what? Even splitting in the presence of two oxygen. Similarly, pyrolase. Pyrolase again, it helps in even splitting in the presence of two oxygen molecules. And the liver enzymes, they are again, this is, for example, I'll give you one, this retinal dioxygenase. A retinal dioxygenase is the enzyme which is required in the degradation of this beta carotene. Beta carotene, or you can say beta carotene, is the major pigment present in the carrots. This beta carotene by this retinal dioxygenase gets converted into, it has 16 carbon atoms, into even splitting. Here, this is a very good example. You can see that 16 carbon atoms can be converted into HC and HC. This is in the presence of retinal dioxygenase, or you can say vitamin A, retinol, retinoic acid, which are the three interconvertible forms of this, are produced by the action of this retinal dioxygenase. So, out of these four options, cytochrome P450 is the example, ideal example of monooxygenase. Cytochrome P450, it is a very important detoxifying agent. It helps in the conversion of, you can say, toxic into non-toxic ones. Whether they are the drugs, whether they are the other foreign chemicals which are entering in our body. So this is basically acting as a detoxifying agent. This is again a very important question. What does this P in cytochrome P450 stands? This P stands for pigment, which means this pigment absorbs at 450 nanometers. So this is a very, very important detoxifying agent. It can detoxify by so many other, hydroxylation is there, acetylation is there. These are the different mechanisms by which that foreign chemical, when it enters in the body, it combines with this, it is degraded to form water soluble products which are excreted in urine. Other detoxifying, if we talk about other detoxifying ones, glucouronic acid is again a very important one. This glucouronic acid undergoes conjugation, which is basically the most important precursor, plays an important role in the degradation of heme. Or you can say it helps in this degradation of bilirubin also, forming bilirubin diglucouranoid, which further forms the bile pigments. So monooxygenases means there is incorporation of oxygen but only single oxygen. And finally it is going to form carbon dioxide and water. So the answer to this question is Cytochrome P450. The next question, ubiquitin is the key protein involved in. Now see, this ubiquitin is what? It is basically a protein and it helps in the degradation. It is clear from the word itself, ubiquitin means it is ubiquitous in nature. It is present everywhere. Ubiquitin helps in protein degradation, It also helps in promoting or preventing protein interaction and another important feature about this is it can affect the cellular location of the protein. Overall, it is degrading protein and it is a very small protein which has approximately 76 amino acids and it catalyzes the degradation of proteins by three major steps. One is activation, other is conjugation and third is ligation. Activation is mainly in the presence of ubiquitin activase enzyme. Conjugation is in the presence of ubiquitin conjugase enzyme and ligation is in the presence of ubiquitin ligase enzyme. So basically it is helping in the degradation of proteins and it plays a very important role in viral infections. It plays a very important role in the transcription of DNA. In DNA repair mechanism, in cell cycle, and it also plays a very important role in the biogenesis of ribosomes. Biogenesis of ribosomes. So ubiquitous protein which is basically helping in the degradation of proteins. Now another very important feature which I would like to bring here is there are three organelles which are involved in the synthesis of proteins. One is obviously the ribosomes because ribosomes are also known as the protein synthesizing machineries. Second is the Golgi apparatus. Golgi apparatus which helps in the synthesis and packaging of the proteins and the rough endoplasmic reticulum. These are the three very important organelles which help in the synthesis of the proteins, which help in, you can say, synthesizing, packaging and then sending it to the their target organelles. For example, you can say synthesis of tropocollagen, synthesis of collagen, synthesis of insulin. These are synthesized by a very important process known as signal transduction. Whenever a cell receives a signal that this type of protein is synthesized, all these three organelles are actively involved in it. So here the answer to the question is ubiquitin, which is involved in the degradation of proteins by either activating it, by either conjugating it with rest of the molecules, or by ligating it. Translation, all of you know, it is simple translation of the information from RNA to proteins. Protein activation, there are so many methods of activation of proteins. Glycosylation is attachment of glyco, means carbohydrate moiety, to a protein. So the answer to this question is protein degradation. Next question. Acetyl coenzyme A can be directly converted to the following except. Now see, this acetyl coenzyme A is the acetyl coenzyme A. This is the most important intermediate of, in fact, all the metabolism. See, whether we are having carbohydrate in our diet, whether we are having protein in our diet, whether we are having fat in our diet, ultimately they have to be converted into acetyl coenzyme A, which has 2 carbon atoms. This 2 carbon atom will be degraded via TCA cycle or given out in the form of carbon dioxide. All the reductant will be there, which will be giving protons. They will be transported via ETC to finally form water. The entire thing is known as biological oxidation. So this acetyl coenzyme A plays a very pivotal role in intermediary metabolism. Carbohydrates, it's very clear, they can be directly converted into acetyl coenzyme A by glycolysis and by oxidative decarboxylation. Proteins, if they are ketogenic, they will be converted into acetyl coenzyme A, and fats, even chain fatty acids will form acetyl coenzyme A. Odd chain fatty acids will form propionyl CoA, which will be converted into succinyl coenzyme A, or some of the intermediates of TCA cycle. So we can say this is very important. And this acetyl coenzyme A can be converted to form, it is converted to form ketone bodies. It is converted to form cholesterol. Precursor of cholesterol and ketone bodies is acetyl coenzyme A. Similarly, acetyl coenzyme A can be converted into fats. Precursor of lipogenesis is acetyl coenzyme A. All can be converted directly into this. Now the question is, can be directly converted to the following except. See, fatty acids made can be directly converted. Just now I told you lipogenesis, cholesterol, ketone bodies, glucose. Acetyl coenzyme A cannot be directly converted into glucose. So the answer to this question is acetyl coenzyme A cannot be converted into glucose. Yes, another question is directly. Indirectly it can. Directly, no conversion. Conversion of acetyl CoA to glucose. Indirectly, yes. Indirectly, yes, by gluconeogenesis. But again, in gluconeogenesis, if we talk about this, there will be involvement of pyruvic acid. This acetyl coenzyme A will be acting as a allosteric modulator. allosteric modulator or activator of conversion of pyruvic acid to PEP. So the answer to this question is glucose. Then next, in Lac Operon, catabolite gene activator protein or CAP is responsible for. Now obviously it has to be positive regulation. Now see, first of all we are going to discuss what is this Lac Operon. This is again a very important question which is generally asked in the exam. Operon is nothing but a collection of genes which is basically required for the regulation and expression of a particular gene. If we have to draw an operon, it's like this. It has a promoter gene, operator gene, a regulator gene and the structural genes. A regulator, operator, promoter and structural genes. Now if we talk about the Lac Operon has three structural genes, Z, Y and A. Z codes for enzyme beta galactosidase, which is responsible in the conversion of in the breakdown of lactose to glucose and galactose. Y codes for permease, which helps in the uptake of lactose, and A stands for trans-acetylase, whose function is not clear, but still we say that it helps permease in helping in the uptake of this glucose. Now see, what happens is if lactose is present, obviously what will happen is promoter gene is what? Promoter gene is having promoter sequences. Basically see, what is happening inside this is gene regulation हम कभी भी बात करते हैं तो it's what? There will be transcription with the help of messenger RNA. If transcription is there, translation will be there, or you can say the desired product will be expressed. Desired gene or you can say product will be expressed. Now if lactose is present in the system, automatically transcription will be there, translation will be there and glucose will be provided to the E. coli. If lactose is not there, obviously no transcription, no translation means no glucose will be provided. So this is what? This is switching on and off of genes. Whenever something is required, genes are switched on. Whenever something is not required, genes are automatically switched off as per the need and desire of the body. Now here the question is regarding this CAP. This CAP is what? Catabolic activator protein. Now here we'll take the two examples. Now see, one is this. This is again an operon. This is another regulator, promoter, operator and structural, regulator, promoter, operator and structural. Somewhere here this CAP is present. Now there can be two conditions. One is if glucose is not there, lactose is there. If glucose is not there, glucose is there and lactose is also there. Now what happens here is, what is the role of this catabolic activator protein? When there is deficiency of glucose, what happens is this glucose triggers the release of the cyclic AMP, which is the second messenger, whose solely function is to activate the protein kinase enzyme. So whenever there is decreased concentration of this glucose, what will happen is this cyclic AMP will be released. This cyclic AMP is going to combine with this cap. When this complex is produced, complex of cyclic AMP and catabolic activator protein, automatically messenger RNA is now free to transcribe. Free to transcribe, and obviously there will be transcription, translation, and glucose will be given to E. coli. Similarly, if glucose is there, lactose is also there, increased concentration of glucose will decrease the concentration of cyclic AMP. Automatically this cap complex won't be produced, so there will be no transcription, no translation, or no operon will be there. So the function of this cap is it acts as a positive regulator by binding with cyclic AMP, forming a complex. So the answer to this question is positive regulation. Then next question is true about histone acetylation. Now see, histone acetylation is what? Basically histone is nothing but a histone. It is a basic protein which combines with DNA. Histones and non-histones होते हैं, they help in regulation of gene expression. Histones are 4 in number: H2A, H2B, H3, H4. H1 is a linker DNA. These 4 form an octamer. Out of these 4, H3 and H4 have lysine residues on the outward side. So whatever modifications are there, they are on the C terminal of lysine residue. Now what happens is DNA is having this type of structure. See, H2A, H2B, H3, H4, this is the octamer you can say, and DNA is wrapped around like this. Similarly, this histone, DNA is wrapped around like this. This is known as beads on a string appearance, and it is also known as a nucleosome. So whatever, this is basically a post-translational modification. Once the entire protein is there, then what happens during, you can say, during the expression of certain genes? So acetylation and deacetylation. Always remember, whenever we talk about metabolism, whenever we talk about synthesis, there has to be an antagonistic process for its degradation, always, so that the entire crux remains in equilibrium. Acetylation and deacetylation means addition of acetyl group. Here it is giving out that acetyl group. Acetylation के लिए enzyme requires is histone acetyl transferase, histone acyl transferase, acylation, acyl transferase, and here it is histone deacylase. Right? or it is HDAC. Now what happens is, suppose, let's take this example, somewhere here one gene is present and we have to express this gene. Obviously, whenever some gene is to be expressed, it has to be unwrapped. So acylation will unwrap that particular gene, unwrap that particular DNA, or you can say, so that it can be expressed. Or you can say transcription will be there. Or you can say ki if we talk about the transcription or unwrapping of DNA, that means it has to be actively transcribed, it should be euchromatin. On the other hand, if we see deacylation, in deacylation we are taking that acyl group out. If we are taking that acyl group out, that means we are wrapping the DNA now. If we are wrapping the DNA, that means we are making it more condensed. And when we are making it more condensed, means it is a darkly stained region or it is rich in heterochromatin. So there is a continuous process, means unwrapping and wrapping of DNA takes place. If the DNA is unwrapped, that means the enzyme is acyl transferase or HAT. When DNA has to be wrapped back, it is histone deacylase. Whatever acyl group we have attached to it, just to expose that particular DNA so that active transcription and translation takes place, has to be condensed. So histone acylation leads to, simple is, increase in euchromatin. Euchromatin means new chromatin, which is actively transcribed. Hetero means different type of things are there, or it is darkly stained. Heterochromatin is further of two types, constitutive and facultative. So here whenever we talk about histone acylation, it has to be the increase in the formation of euchromatin so that it is unwrapping DNA for maximum expression of that particular gene by unwrapping it. Other processes are also there, like methylation. Methylation is again very important, and these all play a very, very important role in epigenetics. But so far as acylation is considered, it is always, there is increase in the euchromatin formation. And that is what is the answer. It increases euchromatin formation. Causes methylation? It never causes methylation of DNA. Methylation of DNA, as per epigenetics, it is again a normal process ki there is incorporation of the methyl group. It has nothing to do with this acetylation. There is a difference between acetyl group and methyl group. Then next question. Which of the following can inhibit the transport of ADP into mitochondria and of ATP out of the membrane? Now see, this is what is electron transport chain. Electron transport chain. Just now I gave you the brief concept of the intermediary metabolism, in which there is degradation of two carbon atoms present in acetyl coenzyme A to form carbon dioxide. Now this water, basically, see, whenever reducing equivalents are releasing these protons are combining in the presence of oxygen to form this metabolic water via this electron transport chain, which consists of 4 complexes. Last complex is the 5th one, where there is cytochrome, or you can say not exactly cytochrome, simple formation of ATP is there. Let's put it that way. Formation of ATP is there. All the reducing equivalents, whether it is complex 1, whether it is complex 2, finally they hand over it to complex 3, then complex 4, which is cytochrome oxidase, forming water. Whatever energy is released at every step, it is utilized in the synthesis of ATP. But if we compare brown fat, what happens is this energy is not liberated to form ATP, instead it is dissipated in the form of heat. Dissipated in the form of heat. Or if we put it in simple language, we can say that this electron transport chain helps in the synthesis of ATP to undergo biological oxidation. Whatever energy is released during the transport of these electrons, it is finally utilized in the formation of ATP to form this ATP. Now there are two different concepts here in this. One is inhibitors and the uncouplers. Let's take inhibitors. Inhibitors, again it is clear from the word, they are inhibiting a particular complex. Whether it is complex 1, whether it is complex 2, whether it is complex 3 or whether it is complex 4. Uncouplers means they are uncoupling a particular process. Means this process, it is uncoupled. Or you can say uncouplers are the ones which won't let the synthesis of ATP. Or the energy will be liberated in the form of heat. Or you can say how these uncouplers act. They can either reject the transport of ATP, either transport of ADP or ATP, क्योंकि precursor of ATP is ADP. So we can say, for example, common example I'll give you here. Let's see the question. Question is, which of the following can inhibit the transport of ADP into mitochondria and of ATP out of the membrane? That is, they are what? They are basically not allowing ATP to form, or they are, you can say, forbidding. They are inhibiting the transport of ADP and ATP. So it is what? It is eryctoside. If we take rotenone, rotenone is what? Rotenone is an inhibitor. Oligomycin is also acting as an inhibitor क्योंकि it is inhibiting oxidative phosphorylation also, but it is not inhibiting the transport. So the answer to this question is A, loside. There are so many questions which have been asked on this. Eryctoside is there, then uncouplers. Uncouplers में सबसे important uncoupler is 2,4-dinitrophenol, which basically dissipates energy in the form of heat. It is also used as, weight reductant drug. So the answer to this question is this. Here one more point I would like to bring, कि cytochrome oxidase is inhibited by cyanide. This is again a very important question. Cyanide is an inhibitor of cytochrome oxidase A1 and A3. So here we finish up with the discussion of this Grand Test 179. I hope I was able to justify the explanations. Thanks a lot.